So we've reached question 5, and we have the essay question. I'm not going to write the whole essay. The electron micrograph shows part of a plant cell, and organelles A, B and C in association with F are all involved in the production of the secretion shown at P. So P is actually going to be a protein which might get secreted out of the cell there by endocytosis, which you'll know about in the membranes topic. However, this is about cell structure. So a good place to start here is let's identify everything. So we've got Golgi body, Golgi apparatus for A. B is our mitochondrion, and you should have seen enough micrographs and textbooks and to see these are actually mitochondria as well. That's just a kind of side-on view. C is our kind of stringy thing with dots on, so that's going to be our rough endoplasmic reticulum. Okay, and we can ignore everything else on that. So those are the names of the things, and our kind of first area that we can talk about is, well, we're making a protein that's secreted out of the cell. That's the story that's going on here. We're making a protein using some of these organelles and secreting it out of the cell by exocytosis. We have got mitochondria there that are powering this process, ATP. Okay, so our first stage, we're going to talk about our rough endoplasmic reticulum, and we're going to talk about ribosomes on there, and we're going to talk about protein synthesis. So the fact that these are kind of membranes with ribosomes on them, but also they can kind of fold, create little pockets called cisternae. And so we're really, describing ribosomes, describing these cisternae, and ideally give an explanation of what's going on. We're getting amino acids, so amino acids, and we're putting those together into a chain, which is going to be our protein. Can we mention compartmentalization? Well, these folds, these cisternae, are going to create little compartments of protein which are going to be separate from the general cytoplasm, and these are eventually going to make vesicles. So that's kind of one area. Our second area that we might talk about, we could talk about the Golgi, and you've used this earlier on in the test. So, for example, modifying proteins. So we can describe the Golgi, we can describe the stack of membranes, we can give its general role. We could say what it's doing to this chain. It's amending this chain, might be adding bits or taking bits away, putting bits together. We can say how vesicles are fusing with the Golgi, so our compartmentalized vesicles containing the protein fuse. So use that word, fuse with the Golgi, and eventually they're going to bud off. So there we are. So budding off, containing a modified protein, and they're going to then move to the cell membrane for secretion there. Why would we want to compartmentalize anything? Well, this protein might be an enzyme, and so if we just have an enzyme floating around in the cytoplasm, it might be digesting organelles, digesting the nucleus, creating all sorts of havoc. So we can mention compartmentalization there as well. And then we have got our last little area where we're going to talk about what have mitochondria got to do with this process. They're not actually involved in the direct mechanics of making the proteins, but we can talk about, well, it's supplying ATP for this process. This process needs energy, so ATP energy. And we can talk about the structure of the mitochondria. So you know all about the structure. I'll do a better diagram than that. Outer membrane, inner membrane folded into cristae, intermembrane space, matrix, that sort of thing. And you can talk about, well, this, we're asked to talk about compartmentalization. Well, the matrix is in a separate compartment to the general cytoplasm, so for chemical reactions there, and supplying ATP. We could talk about our inner membrane being folded to provide large surface area. So for 9 marks, we wanted that entire story in kind of 3 different areas. Most of it is directly, you're just putting the stuff that you know into some sort of order to tell a story about making a protein and it being moved out of the cell.